deliver his keynote address. Sir.
Thank you. I think I share কৰিব লাগিব ৰবা।
Before delivering his lecture, I request Bezbaruah sir to start the technical session by lighting the lamp. Sir, please.
Namaskar everyone. It is indeed a great pleasure and honor to be invited as a keynote to the international conference organized at Nowgong College Autonomous. My sincere thanks
Okay. Good morning, everybody. So, yes, thank you very much for the nice introduction. So, my name is Achintya Bezbaruah. I'm from North Dakota State University. So, what we'll do today in the session, we'll try to confine the keynote addresses to 15 minutes, around 15 minutes. So, I'll be short here, as I wanted to. We can talk more. I am here for 2 days. We can always talk more. I just want to introduce one of the topics out there. I know it is Dr. Sharma. I know it is too early in the morning. I initially thought it's too late, but it's already I think 4:15 or something in UK. So you are awake already. So I thought of asking you to go ahead of me, but looks like as you are awake, it is morning already. So let's do the as scheduled, okay? And welcome all of you who are online. So what we'll do, we'll try to finish the first 3 keynote addresses within the stipulated time. Then we'll talk about the timing later on after this over, timing of the other presentations. We'll try to see how we can finish it. In case we spill over, we figure out a way to have that session after lunch also. Figure out that. Okay. Now with that, I'm going to talk about a little bit about a few technologies that we have used in agriculture. I'm not going to talk about drinking water or anything at this time, but there are a lot of things we can talk about drinking water. But this is what we do. I'm originally from near Kaziranga, place called Bokakhat. So we used to have a lot of land holdings. So we used to do rice cultivation. So rice is the primary thing. So we used to do others also, but rice is the primary one. So this is something I'm very fascinated about. Even though I'm a civil engineer, so my fascination is always towards, can we do something in agriculture? So that's why we started working on that. So one of the things we used to do, among other things, we used to use kosu or taro in our agricultural field. When it is ready to be plowed, so we normally carry a lot of black varieties of the kosu, not the white one, black varieties of the kosu. Then put it in the field. Also, along with that one, we used to put posotia. Posotia is another interesting, this is called vertex. So posotia, another insecticidal properties it has. And then we also, of course, used to put cow manure, gobar. So those are 3 things typically we'll put, and that really gave us very good, result. We didn't know at that point what we are doing, and of course, when I had the opportunity, I had the unique privilege of working in research to find out what is exactly happening in that area. We started some work to find out. So let's start talking about cow manure. What we traditionally use cow manure, so or gobar. So typically, and this is one of the interesting problem. If you look at world over, world is going to run sort of phosphate fertilizer in about 2033. So as you know, nitrogen, phosphorus, and potassium, NPK, the 3 ingredients that we need for our agriculture. So if you have less phosphorus, then we are in bigger trouble. So we have to find out. But other interesting part is nitrogen, manure, which are like a urea and others. Those can be produced from the air. Air has 78% nitrogen. It's a dirty process, very dirty process, not environmentally clean, but still we can produce. We have infinite amount of nitrogen, so 78%. But we do not have infinite number of potassium K, and phosphorus, P. So those are mined like a petroleum. We don't never realize that. So this is exactly what is happening. Not too many people talk about, depleting an amount of phosphorus. So phosphorus is going to last anywhere from 50 years to 200 years, depending upon how we use it. And India or China are the most, uses the most of phosphate fertilizer at this time. And we do not produce it, by the way. So given that, cow manure definitely serves a very good purpose. As you can see, each kg of cow manure, raw cow manure, contains about 1.2 grams of phosphorus, which is a very big amount. You may not, doesn't look like very big amount, but it's a very big amount. Cow manure has a lot of other things, like it has fibers, then we have potassium also. And as you can see, NPK is always there. Also, you have the micronutrients like iron, potassium, and others. Those are very important for plants. Now, cow manure as such is good, but if I ask you right now, how many of you will be comfortable using cow manure as raw? Because our societal change and perspective change, I possibly will not mind touching cow manure right now because I'm used to it. But possibly a lot of us in this room possibly will not feel comfortable touching it. I think this is where we need a little bit of innovation. This is where we need to think about how can you make things acceptable. I understand that many people will promote this idea that we should stick with our old traditions. It's good if you can stick to it. But however, there are over time, things changes. You have to change at the same time, but keeping your roots. You can still be in touch with your roots, but at the same time, you can transform certain things, make it acceptable to society. This is where one of the opportunities come about creating biochar. Biochar is nothing but when you burn something, half burn something, not totally burn it. If you half burn something, then it doesn't become ash. It is not cyanohoinazai. So then basically, it's important that you burn it to halfway through, and then what you got is a biochar. Biochar is, if anybody has seen activated carbon, is like activated carbon.
Slides are moving Google Meet. Can I see?
Yes, sure. Go ahead. Yeah. Okay. Go ahead. Apparently, sites are not moving in Zoom. So I think you shared it, or maybe you didn't share it possibly. Yeah, now share it. Yes. You can say share the computer itself, okay, whatever. Yeah. Now this. Okay. Now do you think they will see it? Can you see it now?
Yes, let me check, sir.
Okay. Okay. Changing.
Yes, it is changing.
Changing now. Okay, good. Okay, good. Okay.
Thank you, sir.
Yeah. Sorry about those who are online. We didn't realize that slides are not changing. Just to recapitulate what we have talked about, we talked about converting, cow manure to more acceptable form across the world is a biochar, which is basically burnt cow manure. So if you burn them at controlled temperature, as you can see on the right side, 400 degrees centigrade, 600, 800 degrees centigrade, depending upon the temperature, you produce different products. So also you can extract a lot of useful manure. I don't want to spoil your morning, but at the same time, if you take human, waste, human poop, and if you do pyrolysis of that one, we can extract a lot of important oil. That oil that is, you're not going to consume them, but you can use it for something else. Those oil can be used for lubrication and others. So that's a very interesting area because, take for example, yesterday we went out for a party. Yesterday we had a lot of food which had oil content. Human body cannot consume all the oil content, but those are very useful resources. Those can be recovered. Same thing can be done from cow manure also. There will be a lot of byproducts coming out from there. So what we did is we produced a lot of biochars from different sources. And what we can do, you can develop modern fertilizer using that. So what we did, we not only use the biochar, we also fortified that with iron. And also we use a particular polymer called chitogen, which is a natural polymer, chitogen. So we made small bits. Like if you have seen superphosphate fertilizer, which looks like granules, we made granules like that. And these polymers are biodegradable. So we looked into those. So these are very nice looking. Like this looks like, pearl or something, very colorful one, and had different compositions. So what we tried to do, we tried to impregnate this beads with phosphate. So if we do that, so we are looking at how much phosphate it can absorb. So it can really absorb quite an amount of phosphate depending upon the composition you have, what type of polymers you're using, whether it has iron, because iron and phosphorus combines together very easily. That's what you find in nature. So now we have also done some plant studies using this. So we did some lab scale studies. So also we did some agronomic studies in the field where we were able to use this fertilizer to supply phosphorus as well as iron to the plants. That's an interesting thing. So we have been able to fortify. In fact, one of our work got a cover page in one of the journals because we're the first group of people to use nanomaterial to supply phosphorus and iron together to plants. Now, this is the other one, the Posotia we talked about. Posotia is a very interesting plant. A lot of you must have had, Posotia is typically used for, with meat and fish. You can actually make a curry out of it and very tasty. I don't know, did any of you had Posotia, Joel? Yeah, many, yeah, okay, yeah. And also you can use actually, we had cows, we had gohali. So in the evening, we'll put like smoke and we'll put some Posotia leaves into that one, and that stays away the mosquitoes from the gohali. It's a very interesting insecticide, and very benign at the same time. And when we looked into details of this one, there are two varieties of Posotia. One is the white with white flower, other is the blue flower. Both of them work equally well. And there are a lot of papers published already on Posotia. I didn't realize that when you're doing things in Bokakhat, we're doing without knowing anything, But these are traditional knowledge that have been passed on to us by others. Now, if we look at the composition of that one, if you look at, some of the polyphenols and others, you have polyphenols and all, those are really, they are antioxidant, and they are also protective of plants, So we do not know yet. We are doing some work right now, how they bind to plants and how to bind to soil and others. We are looking into that one. But this is what we have. You already have a lot of nutrients present in there. This is good in a sense. We used to regret when, Punjab was supplying rice to us. We're thinking that why shouldn't we do green revolution? In fact, we did a very good job. That doesn't justify we being slow, a little bit as a, we are told that we are land of lahe lahe, but that doesn't justify that. But I think at the same time, I suppose important thing is we did a very good job of not embracing green revolution. I think we are in a better position right now to go organic in a lot of cases. This is where traditional knowledge and others can really help us, So now let's talk about, I'm looking at a clock, so let's talk about, this taro, kosu. So now kosu also contains, calcium oxalate. That's normally you give the irritation to you. That otherwise you have calcium, phosphorus, fibers, and starch. So it gives a lot of things. We are doing some work right now. My results are not ready yet. We're doing some work to find out how they bind to soil. If you keep them under anaerobic condition, like in the agricultural field, what happens to them and how they help, whether they get converted to a plant uptakeable stage or not. So this is interesting result. And this is something, so this is what, these are not very sophisticated research. This, anybody can do it anywhere for that matter. And that's what these are low cost, high impact research. So that's the important part about it. Now, of course, let's talk about a little bit about what you can do based on this. What we have been doing, interesting thing, we have been using regular phosphate rock, rock phosphate. And we're coating them with layers of chitogen, which is a biopolymer, biopolymer. And we are just learning everything from nature. Nature does it all the time. And we are also using a citric acid. Citric acid is present in lemon and all. So, so we can use that citric acid. We are coating them with a layer by layer technology. It's called layer by layer technology to develop slow release fertilizer. This is the detail of the chemistry. So how we synthesize it, I can talk more details about this one. Anybody interested, I'll be more than happy to devote time to talk about this, because many times what happens nowadays, research cannot be done in one person. Research has to be done by people spread across the world. Then only you get faster result. We do not have a lot of time. We do not have a lot of time to solve global problems unless you collaborate to do things. Now, what we have developed very nice coated fertilizers for phosphate delivery, and they work amazingly well. As you can see that these are delivery content, how they deliver over time, sustained. We can do slow release delivery. Typical rock phosphate will not, if you use it in your field, they will not be usable by plants, because plants can only uptake 17% of those. Rest 83% get wasted. So we are trying to increase the efficiency of plant uptake by another 20%. 20% is a big job. You cannot do it possibly, but we are trying that. So one of the students from Assam, he's from Dibrugarh. So he's graduated from Tezpur University, his master's. He's working on that one, Sesha Majumdar. So he's working on that one. We in fact submitted a patent already. And we being from the same part of same Northeast, we being from Northeast, that is helping us a lot in our research actually. So these are things we have been doing. I know there are a lot of interesting other presentations, So I told you that I will cover it in 15 minutes, but I think I took a little bit longer time. So with that, these are different funding agencies we had for our organization. So without funding, you cannot, of course, do research, Now, thanks to Nowgong College for organizing this and giving me the opportunity to share this, So now, if you have maybe a couple of questions, we can take 2 or 3 questions quickly. If you have questions, then we'll move on to the next speaker. Thank you. Anybody got any questions or comments? Yeah, go ahead. Can you? Can I go?
Yes, sir. Converting that cow dung to this hydrothermal you are doing.
Biochar, okay.
Yeah, biochar, sir.
Yeah, the next one, yeah.
So here you are again giving nitrogen, purging nitrogen.
Yeah.
You are carrying out the pyrolysis in nitrogen atmosphere.
Yeah, because you have to do it, because pyrolysis has to be done under because we burn things without the presence of oxygen. So to burn the whole thing, pyrolysis is that you have to burn everything without the presence of oxygen. If you give oxygen, everything will be oxidized. You do not want to be oxidized.
That is what I'm asking, sir. Otherwise, sir, that inherent nitrogen, if I don't burn it in presence of nitrogen, that inherent nitrogen that is present in cow manure will be there at 800 degrees Celsius.
Yeah, I mean there will be still some, because nitrogen because not all nitrogen will be going away. Some nitrogen will be still there. This nitrogen that you see here that you are taking, that's not going to contribute to the nitrogen content of the thing, because this is gaseous nitrogen. This just create an inert environment, nothing else.
Actually, sir, what I did, I just used algae.
Yeah, sure.
For defined purpose,
Sure.
for making nanoflax.
Sure.
And then we pyrolyzed one at without nitrogen,
Okay.
and one in presence of nitrogen.
Sure.
And when we communicated this job, that reviewer commented that at 600, my temperature was 550 degrees Celsius. At 550 degrees Celsius, you cannot have nitrogen.
Sure. Yeah.
But when we did EDX and other elemental analysis, nitrogen was there.
No, when they say nitrogen, maybe plant uptakeable nitrogen, but nitrogen will be still there, because you have this, unless you burn it at very high temperature. So most of the compounds will be still there. It's not going to be converted to without that. So I cannot be very specific about what I had. I can communicate with you later on what we found. I do not have the data with me right now. Yes, sir. Maybe one more question. Does anybody has? Okay. Okay, thank you so much. So thank you everybody once again. So yeah, we can, I suppose, move on to the next speaker. Yeah, I'll give it to you.
On behalf of the organizing committee, we express our heartfelt thanks and gratitude to Professor Achintya and Bezbaruah sir, who has come all the way from US to share his knowledge with us. Thank you so much, sir.
Jagannath Biswakarma, right? Okay, okay. Now we'll have a speaker from UK, Jagannath Biswakarma. So he will be speaking for about 15 minutes or so, and now he'll be introduced. Yeah, thank you.
A very good morning once again to all of you. Respected Chairperson, Bezbaruah sir, respected Vice Principal sir, faculty members, researchers, research scholars, students, and one and all present over here. Now I am going to introduce you all to Dr. Jagannath Biswakarma, who is an eminent environmental scientist. And first of all, he is an alumnus of our prestigious institute, Nowgong College. So Dr. Biswakarma is a UKRI-endorsed global talent environmental scientist in School of Art Sciences at University of Bristol. His primary research focuses on understanding biogeochemical processes occurring in waters, soil, and at soil-water interfaces, which significantly impact water quality and agriculture. He also interested in science policy and communication. Before joining the school, Dr. Biswakarma worked at UAG Swiss Federal Institute of Aquatic Science and Technology in Switzerland. He received his doctoral degree in environmental science at ETH Zurich, Switzerland, and he obtained his Master of Science in Environmental Sciences from University of Zurich and a Bachelor in Technology in Industrial Biotechnology from Bharat University, Chennai. Dr. Biswakarma's research interests lie in environmental biogeochemistry, water quality treatment, and the insertion of science and policy. He combines experiments conducted in a laboratory by applying isotopes, spectroscopy, and analytical methods with computational programs to understand the relevance of molecular scale process in the broader context of biogeochemical cycling of nutrients and contaminants in waters and solid water interfaces. Dr. Biswakarma is also working on a better understanding of science policy that contributes to implications of environmental legislation and regulations. He is passionate about applying science communication tools to describe the complexities of impact of climate change on water resources. Dr. Biswakarma has published his research works in many national and international journals of repute. So now I would like to welcome Dr. Biswakarma to join us. Over to you, Dr. Biswakarma.
Namaskar, everyone. It is indeed a great pleasure and honor to be invited as a keynote to the international conference organized at Nowgong College, Autonomous. My sincere thanks goes to the organizing team for their kind invite. And I must be honest, I'm not sure if I qualify to give a keynote at this August gathering. but here I am sharing my perspective on the topic. I must tell you that this is not a data-driven presentation, but a presentation based on some perspective only. So as the title suggests, I will mainly highlight through my presentation why and how it is important to integrate traditional knowledge system with modern science. And by doing so, why it is important to enhance our ancient roots, especially in the field of agriculture. I am based currently at University of Bristol, UK. So if you have any questions, please feel free to write to me at this email address. So let's start with my beginning. I'm an environmental scientist. I look at cycling of nutrients and pollutants across different ecosystems at a molecular level. And I aim to link those findings with macroscopic processes. So in current times, as we all know, that the globe is under tremendous pressure to provide sufficient food, water, and energy resources. These are just to name a few of the grand challenges. On the one hand, you see here in the slide sectors like agriculture and irrigation that challenge to meet current requirements of food production and water utilization. And then on the other hand, we have an energy crisis to run these sectors. My role in this big scheme of things is very negligible, and I think what I contribute here is to understand how biochemicals across these different sectors move from one another and impact environment and human health. So you must be wondering by now whether this traditional knowledge domain even if fits into this picture. And I'm not lying if I say I wonder the same thing all the time. So I read various reports that are published by reputed agencies. For example, FAO has published in 2009 a report called FAO and Traditional Knowledge, the linkages with sustainability, food security, and climate change impacts. In this report, they mention the traditional knowledge and networks of communities and households can be mobilized to prepare for, mitigate, and manage disasters related to climate change before in national level, there's a report published recently in 2020 by Indian Council of Agricultural Research, where they talk about different management schemes like rainwater, soil and water, pest and disease, horticultural crops, farm implements, fishery and animal sciences. I encourage all of you to go through these two reports and update your understanding on the topic of sustainable agriculture and traditional knowledge. What these reports made me convinced that we must integrate traditional knowledge with modern science if we want to keep our ancestral wisdom alive generation after generation. It is quite important that not only we document them, but also support those knowledge and traditional practices with systematically conducted scientific studies. Only then do I believe we can spread the scientific mindset among the new generation and at the same time safeguard traditional knowledge and indigenous practices. But how easy is it to integrate these two domains? For example, in the agricultural sector, it is a necessity that we integrate traditional practices with the food security agenda. But there are challenges such as climate change, which makes us rethink our current agri practices and possibly hinting us to draw inspiration from our ancient past and to bring some old practices forward. It is now also widely accepted, and I quote that indigenous knowledge is a key to sustainable food systems. A commentary article published last year in Nature magazine wrote, and I again quote, agricultural sciences have for too long ignored traditional and local knowledge about crop plants and how best to grow them. That must change if the world is to ensure future food security. And I can't agree more. So it is time for us, especially for northeastern region of India, to come together to document the tribal practices of farming and agriculture if we're aiming to achieve sustainable agriculture and agriculture practices. So now let's see what modern science tells us. Well, we all know that climate change is the worst influence on every aspect of our daily life. An Oxford colleague published this work in Nature Communication in 2022, and they pointed out 12 climatic impact drivers. They can be divided into two sectors, acute and chronic ones. And if you read through this list, you'll find out all of them or many of them will impact your food productivity, quality, and agricultural practices. Similarly, from my own research also, I can confirm that chemicals moving across different ecosystems also harmfully impact food and water quality. Both climate change and environmental changes can cause troubles in our ecosystems and also cause troubles in soil and water quality. That will lead to fatality amongst living beings and also reduce the productivity of quality crops. Here in this right hand side is a diagram that Swiss Ministry of Environment came up with to highlight different chemicals which are pollutants, can be contaminants, and can be even used as bioweapons, who has lot of potential based on their source, based on their release mechanism, based on their exposure, and based on their importance to the natural resources, they can cause challenges to our existing infrastructure and resource system. So I think what is important here to understand that we need to have a combat plan, a plan that can address these challenges, a plan that can have meaningful local and global solutions to our agri systems. And talking about those plans, what can be those of different ideas? Here I list out few that says traditional agricultural practices intersect with modern agricultural technologies. The first one is seed saving and biotechnology, and I think it's very common to remind ourselves that traditionally farmers, even my father, he saved seeds, the seeds which are best performing ones for the next future plantation. So this practice very much is in alignment with the modern biotechnology techniques such as marker-assisted selection and genetic modification, which aim to enhance crop traits such as yield, disease resistance, and nutritional content. The second one is biological pest control and integrated pest management. And I'm very glad to know that we have Dr. Deepjyoti Bhuyan joining us from Sydney today because he and I started out in the Defense Research Laboratory, Tejpur, looking at antifungal activities of plant extracts against fungal crop infection. So this research was, and still, I believe, is inspired by the traditional knowledge available on medicinal plants. So you can see how we can draw parallels between traditional knowledge and scientific practices. Agroforestry and precision farming. Agroforestry, also a traditional practice of integrating trees and shrubs into agricultural landscapes, helps improve soil fertility, conserve water, and provide additional sources of income through timber and food production. And I believe you all know that our tea gardens are the prime example of these practices. Modern precision farming technologies such as GPS-guided tractors and drones complement agroforestry, right? So there are things happening in and around us. We just have to pay attention to the details and follow what is best for our society, for our environment in coming years. Then we have crop rotation and soil health monitoring, which is very similar. I think there's nothing new to here that we alternatively plant legumes or cereals just to help replenish soil nutrients and control pests and diseases. So modern soil health monitoring system and technologies also include sensors and remote sensing satellites that can provide real-time data on soil moisture, nutrient levels, and microbial activity. So by combining traditional crop rotation with data-driven soil management, farmers can optimize nutrient cycling and minimize soil degradation. Water harvesting and drip irrigation. So that's traditional water harvesting techniques such as building, for example, dams and contour trenches, which can capture and store rainwater for agricultural use during dry periods. Modern drip irrigation systems, however, deliver water directly to the roots of crops, minimizing water wastage and improving water use efficiency. So by integrating additional water harvesting with drip irrigation technology, farmers can mitigate water scarcity and sustain crop production in water-stressed regions. So these are just some food for thoughts so that we can draw parallels in practices and sciences and implement, if possible, in our agricultural lands. But this only can happen if we inform our farmers, if we educate them, if we better train them. So it is very important that we ourselves aware of these possibilities and then go in field and inform farmers and agriculturalists. So what should I say finally? I think we all know Assam and in fact the entire Northeast is rich in flora, fauna, and also rich in tribal diversity. There are practices, and we should definitely leverage their knowledge and put forth their practices best way possible. I think it is high time that we come out of the colonial mindset. And to do so, we need to put our own region's science and practices first in our agenda. So, some of the questions that we might to ask ourselves: do we have formed a consortium with scientific data? If not, why not? And if we can, what are we waiting for? Do we have institutional mechanisms that facilitate the augmentation of available traditional knowledge? It's such a huge field, but within this field of traditional knowledge, there are different sectors. Like today, we are meeting an agricultural sustainability sector, right? So different sectors, can we augment them together? Do we have institutional mechanism? Can Nowgong College, now upgraded to Nowgong University, come up with that kind of consortium which has a specific agenda to fulfill, to promote traditional knowledge at the same time, backed and supported by data-driven science? Only then I believe we can promote our own science and we can be really ambassadors for our local, vocal for local initiatives. I think I'll be very happy to initiate some of these tasks as well and promote our science rooted in ancient knowledge. But I would really encourage your thoughts and opinions. I think I like this quote by Walt Disney that the way to get started is to quit talking and begin doing. So I'd like to thank all of you for listening to me. I would again like to thank the organizers, the principal of Nova College in charge, Mohinder Sar, Bhuvan Chandra Sutia sir, the convener of this conference, Farista madam, the team, and especially the all PhD research scholars who have been putting their night and day together to bring this conference in a very good, lively mode. So thank you very much. I hope you all have a great success in this conference, and I hope to receive your feedback in this email address. Please feel free to reach out to me, and I'd like to thank you and thanks all my collaborators, partners, funding agencies so far in my career who helped me build this kind of portfolio and research. So thank you very much.
Thank you, Dr. Biswakarma. Thank you. I think I thank you especially for finishing everything on time. I didn't down 10:33. I was about to go down and write. You have 2 minutes left, but you finished on time. Thank you so much for that. That helps us a lot. Thank you for the enlightening discussion. I think thought-provoking discussion. Now, any questions do you have? Maybe two more questions do you want to ask? Any question? All questions are good here. Be courageous to ask a question. Okay, maybe I can initiate, Dr. Biswakarma. You can hear me well, right? So
Yes, yes. Please go ahead. Thank you.
yeah,
Yeah.
yeah. We have not seen you, but is there a way? I am not forcing you to do that, but in case you can
No, we can.
come
We
up,
can.
that will be great. That will be great. Then we can connect, make better
I mean,
connection possibly. Good,
yes. Can
perfect,
you see
perfect.
me? My camera is on. Okay, cool. Thank you.
Thank you,
Yeah.
thank you. So I've got a question now. How do you think
Yes.
the student population, and the junior faculty members can help in the process? We are trying to look at a low cost, high impact research. Can you just elaborate on that one a little bit?
Yeah, I'm totally in alignment with your thoughts because I think we need low cost and high impact research. And I think Northeast, as you know, I think it's like really fertile ground to do those kind of research. Now coming to your question, how junior faculties and students can support in those kind of research, I think the identification of the problems is a key. So I guess Northeast is full of uh, I will not say problems, but challenges that they need to address for the globe. How climate change, for example, impacting their local community, are their soil being non-fertile in last few years. So small initiative like addressing, identifying problems and what they can do about it. So I'm of course aware of environmental agencies in Assam. I'm collaborating myself with Dr. Farisa Madam, Dr. Bhuyan Chandra sir. So we are trying to address local issues with some small initiatives, and I hope students should come forward to take up these challenges because it's their land, their resource, and it's their kind of high impact, low cost research.
Thank you. Thank you so much. Any one more? I initiate the question for you. You should ask one question. Then, if you ask question, the students also feel good. They feel good. Okay, my talk was worth giving. Who wants to ask that question?
And please ask
Maybe
questions because I woke up at 3 o'clock, in the morning. So please,
I think
so please.
very important. I'm coming over to the back side now. Who want to speak? Somebody need to do it. We'll let you have 2 plates of lunch here twice. You can have lunch twice if you ask a question. Okay, looks like I know this is very clear presentation. I know that the hesitation always to ask questions, but thank you so much for your presentation. If you can hang around a little bit, I know Deepjyoti will be speaking now. Deepjyoti Bhuyan will be speaking. So I think we need to talk beyond this
Sure.
thing. I think that's the most important part. What we continue beyond this thing. So thank you again, Dr. Biswakarma,
Thank
for
you.
your time and
Thank
getting
you.
up so early. Getting up so early. Yeah,
Thank you.
thank you. So our next speaker will be Dr. Deepjyoti Bhuyan. So he is from Western Sydney University. So I know it's a very comfortable time for him. It is, I think, 3:30 or something at that time in Australia. So 3:30 or 4 around that time. So I know you're very comfortable out there. So we can ask you to hang around a little bit longer.
Sure.
Thank you so much, sir. So now, for the next talk, I would like to invite Dr. Deepjyoti Bhuyan. I would like to welcome you for this international seminar, and we express our thanks and gratitude for joining us from all the way from Australia. So now let me introduce Dr. Bhuyan. He is a Research Support Program Fellow, International Engagement Coordinator, Acting HDR Director, at NICM Health Research Institute, Sydney, Australia. He joined his institute in 2018 as an ARC postdoctoral research fellow. Dr. Bhuyan's research focuses on the development of therapeutic agents using gut microbial metabolites, also known as postbiotics, and deciphering their molecular mechanisms of action. Developing natural product-based therapies for bacterial and viral disease is another key aspect of his research group. Dr. Bhuyan is also interested in understanding the complex synergistic interactions among various phytochemicals and standard drugs with specific biological targets. He explores how these interactions can be used as a therapeutic strategies against infectious diseases, including and cancer. In 2023, he was appointed as NICM HRI's International Engagement Coordinator to help strengthen the Institute's partnership with India through research collaboration, engagement, policy development, and scholar exchange program. In November 23, he was appointed as the acting HDR Director at NICM HRI Australia, overseeing a diverse group of more than 40 PhD and medical research and MPhil candidates. Dr. Bhuyan has completed his research in University of Newcastle, where he explored the anti-cancer and antimicrobial properties of phenolic compounds and antioxidants in Australian eucalyptus species at the molecular level. His current research at NICM is supported by several government, industry, and university grants, including those from Australian Department of Industry, Science, Energy, Resources, Therapeutic Goods Administration of and Australia and Western Sydney University. He built active collaborations with over 40 international organizations across the world. Before coming to Australia, Dr. Bhuyan worked as a senior research fellow in probiotic food development at Assam Agricultural University on a national agricultural innovation project funded by ICAR. Since the completion of the project funded by completion of his project MSc in applied microbiology in 2010. He has pursued many academic industry-funded research projects for food chemistry, microbiology, and pharmacology in India and Australia. And his research findings are well published in national and international journals of repute. So now I would like to welcome Dr. Deepjyoti Bhuyan for his lecture. Thank you. Over to you,
Thank you
Dr.
so
Bhuyan.
much. Thank you so much. Can everyone hear me okay?
Yes, we can do.
All right, so I assume that that's a yes. Okay, great. I'll share my screen. Can you see my screen, my presentation screen? So that's the presentation screen, right? Not the Google Meet screen. Please let me know if you can't see it. Okay. Thank you so much for the kind introduction. I think it's always very awkward to hear about yourself when someone is talking about your achievements and things, but I really am very grateful for this invitation. And I would like to thank the organizing committee of the International Conference on Traditional Knowledge at Nowgong College, and thanks for the kind invitation to give a guest presentation at this conference. As you all already have known, that I work as a research fellow at Western Sydney University, and I specifically work at NICM Health Research Institute at the university. I actually am very grateful to Jagannath as well and Dr. Biswakarma, and I always am in like awe when he presents because I really like his research, and it was very interesting to hear Professor Bezbaruah's perspective as well and how he's integrating traditional knowledge with agriculture and environment. So it's kind of a mind-blowing, I would say, in terms of the implications of traditional knowledge that you could have in different aspects of environment and health. So I am shifting the gears a little bit here. So I will be talking more about how we are using traditional knowledge, especially Australian traditional knowledge in cancer research. So this is the title of my presentation, and today
Doctor,
we are,
excuse me, Dr. Bhuyan. Actually, your
yeah,
presentation is not visible from here.
yeah, that's what I was thinking because just a second, let me share again. I think I'm getting some errors here. Okay, so is it possible if I share the presentation with you so that you can share from your end?
Yeah, go ahead, do that.
Yes,
Yeah, okay, so I'll share it with the email address that I
yes, yes,
already
please.
have. Yeah, sorry. It's always a bit of an issue. So which email address should I share it on?
Principal Nowgong College.
Principal Nowgong College. Okay. Um, so that's the .org email,
Yes,
isn't it?
exactly.
So will you be able to get what you call Google Drive on that from that? Because the presentation is quite big, actually. The size of the presentation is quite big. So
Just
Dr. Bhuyan, why don't you do one thing? Just unshare one more time, try to share one more time, because that process may take a long time to transfer it to Google Drive and all. Just try one more time.
yeah,
Try one more time.
yeah, I'm trying again.
Sure.
Okay.
I think it's
Can you
coming
see it
now.
now?
Yes, coming. Yes, that's good now. Yes, go ahead. Go ahead. Put in the
Okay.
presentation. What happens? Yes, I think this is good. Yeah.
So can you
This is
see the
good.
presentation slide or
Yes, yes, we
just
can.
okay, great. Sorry, I'm not very used to Google Meet, so it's always like an issue. So okay, great. So this is the presentation title that I was talking about previously. So we are doing in terms of traditional knowledge, we're using how Australian traditional knowledge can be used in cancer research. Today I'll be presenting my amazing PhD student Indivari Dissanayaka's work, and she's online as well. So feel free to reach out to her as well if you have any questions. So she did this work during her master's, and she has published this work as well. So I will link the papers as well wherever possible so that you can read if you are interested. So I'll also give you an overview of all the things that we are doing at NICM Health Research Institute, Western Sydney University. So I lead a research group called the Gut Biotics Lab at NICM Health Research Institute, and I'll be introducing to those members as well. All right, so as I said, I'll be shifting gears. I'll be talking more about health and how we could use traditional knowledge in improving the health outcomes. So these are some of the global and Australian cancer statistics. So according to the WHO, about 24.6 million people will have cancer by 2030 worldwide. So that's a huge number. And in terms of the prevention strategies, WHO also tells us that about 30 to 50% of these cancers can be prevented by modifying certain risk factors. Okay, so there is that gap or there is that opportunity that we could address through our research. It's specific to breast cancer because I'll be talking about how we are using Australian traditional knowledge to improve the outcomes of breast cancer in Australia. So in terms of the statistics itself, about 200,000 people are living with breast cancer in Australia, according to the National Breast Cancer Foundation Australia. In India, it's actually even worse. It's the second leading cause of cancer death in women. And about 1 in 20 Indian women are expected to develop breast cancer in their lifetime. And collectively, the US, China, and India account for 1/3 of the global breast cancer cases. So that number is pretty huge, right? And there are a lot of challenges. Okay, so when we talk about cancer drug development, so there are a lot of studies that are trying to invent new drugs, right? So there was a study recently in 2020, I would say, in JAMA, which is one of the leading medical journals in the world, and they basically said that to develop a drug from scratch, from discovery to the market, it takes about 780 million American dollars. Okay, so that's for one drug, to develop one drug from basically bench to bedside, that whole approach. It's a huge amount, right, to develop new drugs from scratch. So there should be some alternative, right? So our research group is basically looking at those alternatives. So we are trying to repurpose what we have. So we are basically, what we are doing is that we are combining natural compounds based on the traditional knowledge with standard drugs and trying to basically increase the efficacy of standard chemotherapy and reduce the side effects. Okay, how we are doing that? Before we go there, let's talk about wellness or health. Okay, so when you go to your doctor and if you ask for like a way of living a healthy life, what is the way of living a healthy life? They would tell you three things. They will tell you you eat well, sleep well, and exercise. Those are the three health trinities in terms of wellness. But it is actually much more complex than that. And many people, even if they follow all these things to the point, they won't be able to have an ideal health, what you call status. Okay. So we think there is that, that puzzle is because of the gut microbial communities that are basically living in our digestive system. So these are trillions of bacteria, fungi, viruses that are living in our gut. And those are the keys to wellness. That's what we believe, okay? Um, so why we are looking at Australian um, in terms of the diet and other things, there are few key problems. Okay, so when I talk about from the Australian perspective, about 92% Australians do not eat their daily recommended vegetables. In case of Indians, it's even worse. Indians don't eat, 98% of Indians don't eat enough fruits and vegetables. Okay. And in Australia, we spend about 195 billion dollars every year on our healthcare system. Whereas in India, it's even worse. It's about 372 billion US dollars that was spent in 2022. So those are big numbers, right? And there's a correlation between that because many people are not eating healthy and they're not eating enough fruits and vegetables. There's an increased burden on the healthcare system because of the chronic and other diseases, okay, and metabolic diseases. All right, so let me introduce you to my research group. This is the research group that I'm currently leading at NICM Health Research Institute. We are about 13 members, and we call this lab the Gut Biotics Lab. So you could see that I have a lot of masters and PhD students who are working on different aspects of traditional medicine, gut microbiome, and cancer. So our philosophy is very simple. It actually reflects on the philosophy of Hippocrates. We think that our health begins in the gut. So we are looking at how cancer is different, what are the complex processes of cancer and the gut microbiota, as well as how Australian native fruits fit into that equation. Okay, so it's a lot of different fields coming together, which basically comprises of our scientific curiosities. We are looking at something called postbiotics. Okay, that's like a future of gut microbiome research. So basically, what happens is that when you eat dietary fiber, food that are healthy, like fruits and vegetables, and probiotic, like yogurt and dosa and idli and all that. So when you eat all those things, that will basically go to your gut, and the gut microbial communities will digest that and produce something called the postbiotics. So basically, the health outcome that you see from foods is through postbiotics. Okay, these are the chemicals that are produced by the gut microbiome, which will benefit your health. They will interact with the immune system, they'll interact with your nervous system, and basically they will work together to improve your health. So obviously, if you're not eating healthy, you're eating junk food, you're eating fried food, that's gonna produce a lot of bad postbiotics in the gut. So if you want to learn more about postbiotics, there are a few articles that I wrote for some of the magazines in Australia, and you can read them there. These are the articles. So I'm going to show some of them. Some of them were even featured in BBC as well, because this is kind of the future of the nutrition research, as you can say. So the reason why we're looking at Australian native fruits and vegetables, because they're unique. So like Australia is an island, right? And the animals that we have in Australia are very unique, like kangaroos, koalas, and things like that, where they are very different from the rest of the world because we are an isolated island. So there's like, the biodiversity is very unique, similar to Northeast India's biodiversity, because it is one of the richest hotspots when it comes to biodiversity of plants and animals. So in Australia, there are different types of unique fruits and vegetables, which are known to be one of the richest sources of different phytochemicals like vitamin C, antioxidants, polyphenols, and things like that. And because the climatic condition, because it gets very hot here and gets very cold as well at the same time, so there's that extreme. So because of that, these fruits and vegetables are very rich in secondary metabolites, which are essential for human health. Okay, for example, fruits like Davidson's plum and Illawarra plum, they are rich in antioxidant and polyphenols. So they're about 5 times more polyphenols than blueberries, which are like the poster child of polyphenols or good health when you talk about fruits and vegetables. So this is why we are actually looking at these Australian native fruits and vegetables. And as I said previously, my student Indivari, she's written this particular paper, which was published I think 2 years ago, about Australian native fruits and vegetables and how they can be implemented by different industries, including the health sector. So in terms of cancer research, I'll give you some overview. I'm not going to go into too much detail regarding that data, but please feel free to read this paper if you're interested. So this is another paper that she published last year. So basically what we did, we looked at a fruit called kakadu plum, which is known as the world's richest source of vitamin C. It has, I don't know, about 1,000 times more vitamin C than oranges. So what we did is that we used these fruits and we extracted them using water and ethanol. And then we tried to test these extracts against MCF7 breast adenocarcinoma cells. So these are basically very virulent cell lines for breast cancer, which were basically isolated from patients. Okay, so what we found is quite interesting. So when we talk about cancer research, there's something called apoptosis. So basically, what it is basically a programmed cell death. Okay, so when you treat different with different extracts or compounds or secondary metabolites or standard chemotherapy, you should be able to see the induction of this particular death pathway, which is called apoptosis. And we actually observed that using our fruit extract, which are kakadu plum water and kakadu plum ethanol. And we had another fruit as well included in the study called Illawarra plum. And that also showed induction of apoptosis, which is a very good sign. This is one of the most desirable traits for most anti-cancer research. And same happened with the generation of reactive oxygen species. So basically, cancer is an oxidative stress related disease. So when you have a high amount of free radicals in the body, there are chances that it can act on the normal cells and convert those normal cells to cancer cells. So what we saw is that using this fruit extract did not increase the oxidative stress in those cancer cell lines, which is a good indication that they are working at a molecular level, not just on the surface. So we were able to identify a number of metabolites or secondary metabolites, I would say, which are also essentially the antioxidants like triterpenoids and tannins and polyphenols in those extracts. So feel free to read that paper if you're interested. Then we looked at another fruit as well, which is called native currant here. And these fruits have been used by the traditional Aboriginal and indigenous communities for centuries in their diet as well as medicine, because we believe that diet can be used as medicine as well. So we basically used this particular fruit called native currant and did the exact same thing. We tested them against breast adenocarcinoma cells, and we found similar results. And this paper is currently under review in the journal called Food and Function, which is a Royal Society of Chemistry journal. So, and hopefully it will be published soon as well. So Indivari is the first author, and she did all this amazing work in the lab. And we found that, native current as well induced that death pathway that I was telling you about, called apoptosis in the cancer cells, and it did not increase the production of reactive oxygen species or oxidative stress in the cancer cells. Then we identified different types of secondary metabolites, including tannins and flavonoids and other kinds of antioxidants as well in the extracts, both in the water and the ethanol extracts of native currant. We have some other projects as well based on the traditional knowledge. So we are looking at how Australian native fruits impact the composition of the gut microbiota, and how that can be implemented for treating diabetes. So we are inducing diabetes in animal models like rats, and then we are treating them with different doses of the fruits, native fruits, and seeing what's happening to their gut microbiota composition, as well as their blood glucose level and blood lipid profiles and things like that. When COVID started, we also started this particular project where we looked at how Australian fruits can be used for COVID-19 treatment. We have established a cellular model of a type of coronavirus called HCoV229E to study different sorts of antiviral drugs in the lab, and we are using the same platform to understand if Australian native fruits and vegetables can be used, whether they can be used for treatment of COVID-19 or related diseases like influenza, for example. These are some of the projects, as I was telling you about, these are that I'm currently supervising. You could see a wide variety of projects from Australian native fruits. So Indivari has started her PhD after finishing her master's, and she is now trying to produce postbiotics that I explained previously from Australian native fruits using probiotic bacteria. So we basically want to produce this postbiotics so that we can improve the health outcome of Australian, as well as meet the daily recommended dietary intake need as well. These are some of our papers from the research group, and it's a wide variety of papers from avocados to immunotherapy to mushrooms and gut microbiota and postbiotics as well. So please feel free to have a look and read those papers. Most of the papers are open access as well, so you should be able to access them openly. Yeah, so overall, food for thought would be, if we can combine traditional knowledge like Australian native foods and futuristic knowledge like postbiotics, we should be able to improve health outcomes in the future. Um, yeah, and this is the lab website. Have a look. Here we basically list everything that we are currently doing at our institute in our lab, and this will give you most updated will give you most updated information about our research and the activities of the lab. And thank you for listening. I hope it was useful and you found it interesting somehow. Thank you.
Thank you, Dr. Bhuyan. I think it resonated with everybody, because now you know why do we take some traditional fruits and traditional vegetables and everything. Now I'm very sure there'll be a lot of questions this time for sure. Okay. Now who want to start? Some questions or experience your share your experience with him. You want to talk about some Assamese food found in Northeast and Assam? You can talk about that also. Who wants to start? You can ask in any language for that matter. You don't have to ask in English. You can ask in any language. I want a really courageous volunteer here. কোনে সুধিবা? নগাঁৱৰ সন্মানৰ কথা আছে, কোনোবাই সুধিব লাগিব এতিয়া।
Yeah, মই তেজপুৰৰ হয়, so I'm expecting more questions from Nowgong.
Yes, sure. Absolutely, yeah. I think there's a competition between Tezpur and Nagaon also. Now you need to ask questions. So, kune khodiba? Okay. Now, Dr. Bhuyan, so you said that this, because of the extreme temperature conditions, you have enhanced quality of the product, right? Enhanced quality of the. Is it true? Okay, that's true. Now, what do you see in Northeast as compared to? I know you must have studied in Northeast. In terms of, can you name a few of the vegetables and fruits that you found in Northeast were potential candidates for similar kind of research? What I'm trying to do here, we just wanted to put some more additional food for thought, such that they can think about traditional items and get started working on them.
Yeah, absolutely. Very good question. I mean, I was very intrigued to see when you mentioned Posotia in your presentation, because we did our research, as Jagannath was telling previously, we did our research on Posotia back in, I think, 2010, where we looked at how that can be used for the antimicrobial properties, basically, antibacterial and antifungal properties as biofungicides or biopesticides. So that is very interesting. In terms of foods, I think Northeast is like so rich in there's the fruits and vegetables that we traditionally have are quite rich in secondary metabolites, similar to Australian native fruits, I would say. Off the top of my head, I would say that, from speaking from a gut microbiome perspective as well, I would say that including fruits and vegetables in the fermented form would be better in terms of health outcomes. So basically, because they will increase the bioavailability of these foods. So as I was saying before, even if you meet the dietary recommendation by WHO or FDA, most of the times what happens is that you eat all these fruits and vegetables, but your body is not able to absorb that because of the low bioavailability of most fruits and vegetables. So one way of doing that would be to ferment them, right? We have things like bamboo shoot fermented, like gas and all. So those are very good for sources of fiber as well as probiotics and secondary metabolites and antioxidants. So bahar gas is a very good example. Off the top of my head, I'm just thinking, and traditional foods like otenga, leteku, those are very good as well. I know that they're disappearing because people are much more interested in having like Western fruits like apples and grapes and things like that, these days, rather than looking at the traditional foods that we have. So I would highly encourage people to eat the seasonal traditional foods. And Assam Agricultural University actually does an amazing job doing that, because they're trying to basically promote these fruits and vegetables and basically processing them in a way that that can be utilized by the wider community. Because most of the time what happens is traditional fruits and vegetables are perishable as well, that because of the lack of post-harvest technologies, where you cannot preserve them for a long time. So in that case, we can use traditional practices like fermentation, like pickling, for example, is a good way of preserving those foods, and also to increase their bioavailability so that you get the most benefit out of them.
Sure. No, thank you. I think that's gives us. I got a question next year.
So thank you for your amazing presentation. Yes, I have a question. Like you have only used two solvents for extracting, like ethanol and water. Why not others? Because we know that methanol is most amazing solvent. Because it's edible only because of that reason, or for other?
Yeah, absolutely. I had this conversation with Indivari yesterday, because we are basically arguing. So she suggested that, oh, why not methanol? And because it will be much more in terms of the antioxidant activity, we'll get more. But the idea of using ethanol, To be honest, we don't even want to use ethanol, but we have to be practical as well. Most of the alcoholic beverages that are prepared traditionally by the indigenous communities of Australia, they have ethanol in it. So that makes sense to use ethanol. Water, because of the physiology of human body, because we are 90% water. So basically, that is the whole reason we only wanted to use water and ethanol, and with more emphasis on water, because the idea is that if you use water to extract these fruits and vegetables, and you can use the same methodology for your projects as well. If you're running any projects on fruits and vegetables from Northeast, first preference should always be on water, because that will be more bioavailable to the cells as well, when you treat them with different extracts.
Thank you. I think that's one of the things, I suppose. We do a lot of water extraction because it's so green, right?
Yeah.
I know it's difficult. It's difficult because, yeah, exactly. Yeah. So, no, I think that's a very important point. If you look at simple analogy, forget chemistry that we taught, Dr. Bhuyan taught, Dr. Biswakarma taught, simple thing that you always do at home, when you cook taro, kosu, you always, first, you cook it, then you decant it, throw the water away. That's an extraction of the bad chemicals initially you do, right? The simple things. These are not rocket science. They are simple science we're doing. So one of the things, I think, what came out from Dr. Bhuyan's presentation, I just like to summarize. Do anybody has other question before I summarize this session? So, one of the important things that came out from here, it is not only the hardcore research, but also there's a potential for entrepreneurship, then industry, setting up industries and all. You have a question? No? Okay. Setting up industries and all. So you can, in fact, doing a proper business of pickle can be a good one. Only thing you need to ensure that all the nutrients, micronutrients, polyphenols are preserved properly. That's very important. I think that is something you can think about. You don't have to be a scientist. Not everybody needs to be a scientist and should not be a scientist, rather. So what we have had today in all the discussions today, one of the things, overriding, emphasis is that communication and documentation is very important. So what we can do as such, you can start documenting people. One of the things we have started recently is the documentation of folklores and folk tales. Also you can document. You can do oral. You don't have to write anything. You have a camera, phone with you. You can do what is known as the oral history. You can talk to some of the people, elders, who are very senior. Then you can talk to them. What did you do? What did you use for this particular medicine? Take for example, we still remember sojina plants. Sojina plant, the root of that one, we used to use it for whenever cows and everything has some wounds and everything. We used to use that for healing and taking out the maggots. I think those are a traditional. You can record it. You don't have to be a scientist. You should. Everybody has cell phone. Let's use the cell phone for better purpose. And thank God that you're not using TikTok anymore, which is good. It's good. I mean, in US, we are trying to do that. There's a lot of politics going on out there. But, there is something, there are world beyond Facebook and, or whatever, X, that we should have that. Use the phone to record your grandma's, what grandma has to say, how to cook things, how to preserve things. I think those would be important. Nowgong College can start and, oral history museum, maybe, that can be a very good one. Just record it, because we find it difficult to write things. You're a little bit of, but recording is very easy. You can record it and transfer it to the mainframe. I think that's what it is. Let's thank Dr. Bhuyan for his excellent presentation, and also Dr. Biswakarma here. So we'll take a break, or we just continue. We'll continue with that one. Okay. So with that, we came to the kind of end of the first part of the session. Now, what we'll do now? How long? So we can continue up till what time? 11:30. How many minutes left now? 20 minutes. 20 minutes. Okay. I'm targeting 2 papers to be covered within this 20 minutes. So what we'll do, possibly we'll present it for 8 minutes. 8 minutes presentation and 2 minutes for question and answer.
So 5 plus 3.
5 plus 3. Okay, 5 plus 3. I can do a little bit of adjustment with your permission, but let us do 6 plus 2. 6 plus. Let's present for 6 minutes. I know you came all the way, you prepared everything. Let's talk for 6 minutes and 2 minutes of question and answer. But that 2 minutes has to be properly used. We have to ask question now. Good. So,
Sir, first presentation.
okay. Okay. First presentation we have. The first presentation we have is by Tanmay Sarkar and Sabrin Ahmed, Dr. Sabrin Ahmed, study of traditional agricultural practices in Khasi society in Janisperia's everything the light touches. So that's the one discussion we're going to have. So I think Tanmay is going to present, right? Yeah. Tanmay, all yours. Yeah. So how things going to work is because we are all physically present here. So as it becomes, 5 minutes, I'll walk over there and stand there. 1 minute left. So you know that already. So yeah. You can continue another 30 seconds if you want extra 30 seconds. That means you get 6 minutes and 30 seconds.
Now we have 5 papers.
Huh?
5 papers.
Okay.
So first one is here, and then we call this update because they have arrows.
Okay.
Okay.
So this is second paper.
This is the second paper, third, fourth, and fifth.
We can cover it. As we prepare for this one, our second presenter will be Nandini Priyam Rajkumari, Kalyani Adhikari, and Pallavi Goswami. So there will be a second presentation. We'll try to cover 5 of them right now. And third one will be Bonita Boras, and next one will be Snigdha Nath, Subhasis Das, and the last one in this morning session will be MS Dorothy and others in this presentation. So other people, please get ready for the next, so I can optimize the time. Sure. Do you have a speaker out there that works? Or that works, right? Okay.
Hello.
It works.
Good morning, everyone. My name is Tanmay Sarkar. I am a scholar at the Department of English, Nowgong College, Jagannath, and I, along with my guides, Dr. Sabrin Ahmed, Associate Professor in the Department of English, Nowgong College, Jagannath, we are going to present this paper. Hello. The title of my paper is A Study of Traditional Agricultural Practices in Khasi Society in Janice Pariat's Everything the Light Touches. So, Everything the Light Touches is the latest novel by Janice Pariat. Janice Pariat is an Indian writer who hails from Shillong, and she has been born and brought up between Shillong and Assam. So, this text has mainly 4 narratives. The 4 narratives are those of Shailene, Evelyn, Johann von Goethe, and Carl Linnaeus. Today, I'm going to focus on the narrative of Shai. So, this narrative shows the major contrasts between a fast-paced life and a slow-paced life. The life in Shillong and Delhi versus the life in Maumalang, and the traditional agricultural practices that are practiced in Maumalang, and how it has led to the development of Shai's self-identity and personality. Now, let us look at farming in Meghalaya. Now, about 83% of the total population of the state depends on agriculture for their livelihood. The major crops grown are rice, maize, orange, pineapple, lemon, jackfruit, bananas. All of these food crops are found in this region. Farming practices that are shown in the text. So we see that Shai, through farming practice, has grown from an amateur in these genre to a person who knows the basics about farming, and how this has led to her personality development as a whole. We see that the practice of hybrid farming has been shown in the text. As we can see here, The beans have been planted with the corn, the ginger with the pineapples, and they are growing well together. So we see how hybrid farming has been a part of traditional farming scene, and these are shown in the text. Now, the spring festivities related to harvest are also shown in the text, like the Nongkrem. It is a dance festival related to harvest time during autumn, and Chad Sukra is also a springtime festival which is related to agriculture. Now, about this novel, as I have said that it is about the four characters, Shai, Evelyn, Goethe, and Linnaeus. They are distanced by time and place, but they are united by the central theme of ecological consciousness. That is the main crux of this novel. Shai's narrative is based in Meghalaya, mainly in Shillong and Mamalang. Mamalang is the place where she goes to visit her nanny, Oin, and we see through the image of this character, Oin, how a person is deeply rooted with the soil, and even in the later stages of our life, how we crave for our motherland or the touch of soil. The ecological concerns in the text. Shai's narrative addresses the issue of ecological consciousness. Now, what is ecological consciousness? It is the awareness of our relationship with our environment or with the nature. And here we see the word plant bias that comes up in the text. It is the human tendency to underappreciate or ignore the flora around us. So basically, plant bias is aimed to be solved through ecological consciousness. The use of common lands for agriculture, it is a very important technique that enables us for sustainable practices in agriculture. That is the use of nongkinti. Now, the village council of Meghalaya, it allows many people to share a common land during different intervals of time, which enables a large number of people to make use of the land resources efficiently. There is also mention of acknowledgement from trees. That is, when we take something from nature, we need to acknowledge it. And that is shown in this text. The menace of uranium mining has also been depicted in the text. And when we see these lines, why would we name what we didn't need? To the people from the government, though, it mattered. This has been used here with reference to uranium, which the local people only know as yellow cake. For the local people, uranium has no value because it is only harmful to them, because it has led to many diseases like cancer and even miscarriages in women. So the local people, they are against this uranium mining methods. However, the government, it has been said that the government insists in these practices. The Diengi, or the first tree of the Khasi tribe, is also shown, which can be compared to Goethe's earth plants, or the idea of the archetypal plant. Now we go to the Khasi agricultural practices that has been shown in the text, mainly jhum cultivation, and as I have shared, the sharing of the common land, that is Nongkinte. We have come to the conclusory part. The text has incorporated the essence of traditional Khasi farming, which is that of a sustainable and community-driven approach. Through this text and through this paper, I would like to focus on the comparison that can be seen between the tradition and modern ways of looking at things, and basically how we have treated nature in our past and how we are treating it in the present times. So with this, I would like to conclude my paper. Thank you so much.
Thank you. I think you finished perfectly on time. Thank you so much. Now, this is a very interesting presentation of a novel and how it is connected. So very good critique. You critiqued it very nicely. So now we need to have some questions, one or two questions. I'll request the next presenter to be ready by this time. You can come over to your slides so that we can save some time and be ready for the inaugural function. So who has a question now? One of you is presenting the content now. So you need to ask questions. Any language for any. Have you read the book? How many of you have read the book? So you know what book to read. You read that, okay? You have a question. Okay, you have a question. Okay, sure.
I have no question, but I feel very good to heard that actually my PhD topic, the field was in that uranium mining side. So I feel very nice to hear from them.
Okay. Any other question you can ask?
Professor Bezbaruah, can I ask from online? This is Jagannath here.
Absolutely.
Yeah.
Yeah, go ahead.
Okay.
Jagannath, go ahead.
Thank you. So I have a question for you. It was a very good talk and very. I mean, I'm much benefited. So it's a general question. How often do you see in novels or in writings from Northeast talking about environmental sciences or about food habits?
Go ahead. You can take this one. Yes.
Thank you so much, sir. Hello, sir. Thank you so much for your
Yes.
appreciatory words. Sir, actually, when we say about writings from Northeast, now actually it has become a pan-Indian genre to say. But specifically, if we say about the writer Janice Parriott, in this novel, Everything the Light Touches, we see that the novel is based on ecological concerns. Now, when she has given a comparison between Goethe and Linnaeus's views, she has also tried to present how we are looking at nature in the contemporary times. Like we try to separate everything factually. Why is this happening? The earth is in this condition. Why is this happening? Why not? So while questioning also, we need to keep in mind the context, like where we are and what can we do from this position itself. In the northeastern perspective, I think that Janice Parriott's writings, we can take Sumana Roy's writings, we can take these ecological questions have been come up in these works quite effectively. I would like to say that to you, sir.
Okay, good to know. Good to know. Thank you so much. Thanks a lot, and all the best. Yeah.
Thank you so much, sir. Thank you.
One more question. Can the next presenter come over and are you presenting online? Next presenter or? Okay. Yeah. Can you just set up your? Yeah. So we have Nandini Priyam, right? Nandini Priyam Rajkumari. Okay. Go ahead. Set up your system. And anybody has a second question? Another question or comments? No. Okay. This is one of the art. I know it is little bit. You feel a little bit scared to ask question because this is one of the art. You should be able to ask question, participate in the process. Do you know any Assamese novel that has talked about environment? Yeah, go ahead. Think about a novel which is talked about
এখন
environment.
অৰণ্য আছিল তাৰে ওপৰতে ইয়াত এখন অৰণ্য আছিল actually it was a novel, beautiful novel. You can read that also very nicely.
নৈ বৈ যায় by লীলা গগৈ, where ইমান বহুত Assamese traditional things are well explained in নৈ বৈ যায়. I think every Assamese student should read that.
One of the things, remember, if we do not know about problems in Assam, we do not qualify to call ourselves as Assamese. Okay, just because you have an Assamese name, you speak Assamese, you are not an Assamese. You need to know about the local problems and local traditions and everything, problems or whatever, not necessarily in this area, but something you need to know in that, you should be proud of what you have, or you should be able to identify the grand challenges that we have. Okay, thank you again. Thank you for your presentation. It's a very nice presentation. You spoke very well. Thank you.
Good morning. Myself, Kalyani Adhikari, and my fellow mate, Nandini Priyam Rajkumari. We are from the Department of Chemistry, research scholars from Nowgong College and Guwahati University. Today, we are going to give a seminar on the International Conference on Traditional Knowledge, and we have submitted our abstract on the topic, Synthesizing Nano Anti-Inflammatory Antioxidant Medication Using Turmeric-Reduced Copper Nano. First of all, what is traditional knowledge? It is the accumulated knowledge and experiences of the indigenous people, local communities, and local communities and traditional societies, often relating to various aspects of life that include health, resource management, healthcare, spirituality, and cultural practices. And uh, and for the sustainable knowledge, agriculture refers to the farming practices prioritize sustainability or the ability to maintain the process over time. As our topic is on the curcumin loaded nanoparticle, so we have taken a turmeric which is renowned for its medicinal properties, and it also plays a multifaceted role in sustainable practices, which reflects its significance with deep-rooted tradition and associated across diverse societies. And and the turmeric, it's having a chemical constituent is a curcumin. For the utilization of turmeric in cutting edge in nanotechnology, we have the drug delivery system, which is the curcumin-loaded nanoparticles have been developed in drug delivery system to improve the solubility, stability, and viability of curcumin, where nanoparticle been encapsulated curcumin, protecting it from degradation and enhancing it delivery to target tissue and cells. Then, for the diagnostic imaging, turmeric nanoparticles, which has been utilized or as contrast agent for diagnosing imaging techniques such as MRI, resonance imaging, and fluorescence imaging. Then, for the antimicrobial nanomaterials, turmeric nanoparticles have been investigated for their antimicrobial properties against bacteria, fungi, and viruses, which enhance the antimicrobial activity for combating drug resistant pathogens and controlling diseases. And for the food and agricultural nanotechnology, turmeric nanoparticles have been studied for their potential applications in food preservation, packaging, agricultural products, which can not only enhance the delivery of um, bioactive compounds in agricultural formulation, such as pesticides, fertilizers, and plant growth regulators. So overall, turmeric-based nanotechnologies had promising for a wide range of industrial and agricultural applications. And the next part of the presentation is going to be by the Nandini. Over to you, Nandini.
Thank you. So the next part is copper nanoparticles. So nanoparticles hold significant promise across various fields. Like it has unique properties that has developed the versatile applications. Now the copper particles has these unique properties like antimicrobial properties, catalytic activity, electrical conductivity, and energy storage and biomedical applications, and this makes a various diverse field in applications. Now our main objective of our research is to synthesize plant rich in bioactive compounds. We have taken curcuma longa, which will act as a reducing, stabilizing, and capping agents during the process green synthesis process of copper nanoparticles. And the application is like applications in dressings of bone infections. So for methodology, we have taken copper acetate as our precursor salt, and the reducing agent is our curcuma longa, that is turmeric, and CTAB is taken as a binding agent. So 0.002 molar of copper acetate solution is taken in a conical flux, where we have taken as a copper acetate we have taken as a precursor salt, and for turmeric we have taken that or obtained from Karbi Anglong district, and then we have extracted the turmeric solution. Now the after that we have added CTAB 0.012 gram and stirred for about 10 minutes, and for to remove dissolved oxygen, we have purged nitrogen gas in the conical flux, so that no oxygen can be present during the process. And after that the stirred is done for about 10 minutes, and we have added drop-wise turmeric extract solution, 6 ml, and then we kept for stirring. And after that drop-wise addition of turmeric, we can see a change in color from yellow and green solution. This we can indicate that the turmeric has been can be added as a reducing agent. Now we have done a centrifugations, and we have obtained our copper nanoparticle, green synthesized copper nanoparticles. And for result, we have done the XRD analysis. We have for the synthesis, XRD gives us the structural information of our copper nanoparticle. We can see trees of peaks here in this figure of XRD, which also correlates our literacy survey. And for UV spectra, the copper nanoparticles shows UV range in 280 to 390 nanometer range, which indicates our synthesis of copper nanoparticles. Now this is the same image of our synthesis, green synthesis of copper nanoparticles. It gives us the surface analysis of the nanoparticle. It is cubic in nature, and the average size is of the copper nanoparticle is found to be 41.4 nanometer range. So for application part,
For the application part, we also we done some antibacterial activity to investigate the antibacterial activity. We have some we done well diffusion method where the agar been used for distributing bacteria culture on the agar media plate allow the culture to fully absorb into the media. Then following the agar solidification, each bacteriophage has 6 mm of diameter where the copper nanoparticle being placed and incubated it for 37 degrees Celsius for 24 hours. And based on the presence of the zone inhibition, the results have been recorded and zone size was calculated. So for the conclusion, so overall, we have found that the turmeric shows a mighty sustainable practices, and it is a less cost effective, less time consuming, no noxious chemicals methods are being used, and it might hold for the further, it might hold for the promising industrial production. That's all. We conclude our presentation. Thank you.
yeah.
Okay. Now, this is again another familiar thing. You have it every day, right? Haldi you can have every day. Now there should be questions. So, what questions do you have before we'll break after this? We'll break after this for an inaugural function. The same session will continue post-lunch so that we can finish all the papers. So, what questions do you have? Yeah, go ahead. Thank you.
Thank you very much. It's a very nice presentation. Just I was wondering, just did you do the test without any copper nanoparticle, and do you study any toxicity study? Because like copper, sometimes if you consume it can be poisonous. Do you do any toxicity study so that human can also take that kind of copper nanoparticle in your body? I would not definitely. Do you have any thought about that?
I'll give you this microphone. Yeah, you can use this one.
Thank you, sir, for the questions. So for toxicity part, we the analysis we are going through, but for our reduction process, so the study is still going on. And also HANA we have taken for also we have taken beat studies on progress. Yeah.
Questions.
Have done some antimicrobial activity test, isn't it? So what bacterial and what fungal strain you have taken? Because I think that was for fungal, no? So what fungal strain and what bacterial?
Thank you for the question. So, ma'am, we have taken a bacterial infection, E. coli.
Can you go back to the slide?
No, that's not equal. Like, that green one is, I think it's contamination.
Yes, contamination.
So it's contamination.
Little bit there is some contaminations.
Full contamination. But I think before
Actually,
do it again.
okay, ma'am. Actually, ma'am, we have,
Transition only.
ma'am. Actually, we have did for the first time. This we'll continue for the rest.
This is not bacteria. This is fungus and it's contamination.
Okay, okay, ma'am. Thank you, ma'am.
Can you go back to your sorry, XRD slide? Yeah, previous one. I'll stop here. So one of the things I saw, you asked that question about whether you have done any test. I know your regular analysis of things. Here, what I'll suggest you to do, as you are doing XRD, have you done an XRD for only Haldi? Have you done one to baseline, as a baseline? It will possibly come as a very rough baseline because it will not possibly show up most of the things. That would be a good idea to put them together such that you know what is coming from Haldi, what is coming from your synthesis part, right? That'll be good. Always, even if you're not really comparing the toxicity, at least you need to be able to compare what Haldi is giving to this process, right? That is important. I think that control experiment you have to conduct. Good work, very good work. So you're doing your master's or PhD. You're doing your PhD, yeah. But then we'll be demanding more stuff from you, right? So we'll be demanding. I saw that you're doing master's, then we can demand a little less. But if you're doing your PhD, it has to be thorough. You have to contribute to the knowledge. I think you're doing a very good job, very good start. I think this is what most critically think what other people have not done, right? I think also think about a control experiment, control and blank. Those are very important. Yeah. Thank you for your presentation. Any one more question? Anybody has one more question before we break? Okay. What we'll do?
I have
We'll
a
break
question.
for right now. Go to the inaugural function. Okay. Go
I have
ahead.
a question if you don't mind. Yeah.
Sure. Absolutely.
Yeah, so very interesting presentation, and it's kind of something quite relevant as well in terms of traditional knowledge, curcumin and haldi, and all that. But be mindful of one thing with curcumin is that it is also considered a basically pan-assay interference compound. So what happens is that when you use curcumin for biological systems like bacteria, fungi, or even cell lines, it will interact not very specifically with different targets. So basically, it will show you positive results in different things. So you need to make sure that doesn't happen. So always have, as Professor Bezbaruah suggested, should always have curcumin as your control so that you can see whether it is increasing or decreasing the activity compared to the control. And also when you're I presume that you're going to use it as an antimicrobial agent. So it is very important to choose those microorganisms quite carefully as well. So when you're targeting antimicrobial activity, you should always target a wide range of organisms. It can be gram-negative. I think you said that you are looking at E. coli. Is there any gram-positive organisms you are looking at as well, in addition to fungi and candida, for example?
Sir, thank you for the question, sir. So actually, at present we are taking only the E. coli. Future we can take some other bacteria also.
Yeah, so it's very important to include both gram-positive and gram-negative bacteria, and also include curcumin, standard curcumin, the compound, as your control, because you are trying to, basically the idea is that you are trying to increase the efficacy of curcumin, right, using copper nanoparticles. Is that what you are trying to achieve?
Yes, sir. For the safety and
so in that, absolutely. So in that case, you definitely need a curcumin control, and you need a wider range of microorganisms. Also, the source of those microorganisms is important. E. coli derived from plants can be different from E. coli from humans. So you need to basically target your organisms based on the application that you have in mind.
Okay, sir. Thank you, sir.
Yeah, thank you.
So I think what you said, Dr. Bhuyan, is important. I suppose having controls, I think as a PhD student, I'll ask you to be control freak. You control, because if you're producing some copper nanoparticle, bio-based copper nanoparticle, how about non-bio-based copper compound. How do you claim that your copper nanoparticles are better? You have to have those controls as well. Think about that. It will be time consuming, and that's what PhD is for, right? PhD is for time consuming. You do not know in how many years you're going to get your PhD, right? That's what it is. Yeah. Thank you so much for your presentation. Very nice work. And thank you, Dr. Bhuyan and Dr. Biswakarma, for being here today. So if you can join later on, please do. Otherwise, I know it will be, maybe you'll be get busy after some time. So thank you again. Thank you, everybody. Now we'll have a tea break. I said we have a tea break. Then after that, we'll go to the inaugural function. Then after lunch, we'll come back and assemble the same room. Thank you.
Inaugural function will be here only.
Oh, here only,
So
okay.
please be here after the tea break. Thank you.
আৰু এটা কথা, there is one scanner that is a barcode at the end of your notepad. Notepadৰ শেষত এটা barcode দিয়া আছে. You please scan it. You will get a form. Fill it up. That is a feedback form. To get your certificate, you need to fill up that feedback form.
Hi, is there someone there? I mean, can you hear me? I'd like to hear one of your voice if it's possible. I don't know what's going on. Oh, I don't know.